Welcome to the Pymol tutorial for Bio 320. Just gonna open up my browser here and show you the two different websites that you'll be looking at. So the first website here, this is the Pymol Wiki. It just gives you some more information in case you want to make your assignment figures look a little bit nicer. It's completely optional, just follow what's in the tutorial though. You don't need to go into this page if you don't want to. This is the PDB file website, so it has the protein data bank. It has all the different 3D structures of the proteins. So we're going to take two proteins that we're going to be looking at in this tutorial, and we are going to download them. So the first one that we're going to download is 3SPU. So we're just going to click that. And this is the crystal structure for New Delhi metallo-beta-lactamase. So we're just going to download the files, go to PDB file and text, and we're going to download that. It goes directly to your downloads folder. The next file we're going to download is 1KO3. So 1KO3, go download files, PDB text, and we're done. So we downloaded our PDB files. We're now ready to start working with Pymol. So I installed Mac Pymol. I'm just going to open up the program, and we're going to open up our different molecules we just downloaded. So we go to Downloads and go to 3SPU, which is the one we're going to be working with. So this is the three-dimensional figure of 3SPU. It's currently in lines right now, so you can see it's pretty messy. It's kind of difficult to tell what's going on. We're going to change that in just a little bit. But first, let's go look at the amino acid sequence of this protein. So that's very useful to have. So you'll notice here that there's an S button here down at the bottom right of the Pymol window. We're just going to click that, and you can notice now at the top of the three-dimensional structure of the protein, you now see the amino acid sequence being shown, one-letter abbreviation, so that's why it's important to know them. Alright, the next thing we're going to do is we're going to go over the control panel. We're just going to go over the A, the S, the H, and the C button, and we're going to go through those step by step. So right now, as I said, this is the three-dimensional structure of beta-lactamase in lines. We don't really find that all too useful. So one of the things we're going to do is we're going to make it into cartoon mode, which basically shows you all the secondary structure elements of the protein. So under the control panel here, when I have 3SPU selected, I'm going to go Show and as Cartoon. So now you can see this is a lot better looking in terms of the three-dimensional structure of beta-lactamase. You now see the alpha helices, the beta sheets, the beta turns, all that kind of stuff. And that's quite useful. So next thing we're going to do is we're going to just look at the A subunit of beta-lactamase. So the A subunit, you can see that's at the top here. This is representative A. If you scroll down all the way to the right, you eventually get to the B subunit. You keep scrolling more, you get to the C subunit, but we're not really interested in that. We just want to look at the A subunit for this tutorial. So to highlight the A subunit, it's a little bit tedious. You need to go through the entire highlighting process. So you just left click and you drag all the way to the right. So we start off at all the way from aspartic acid 43. You can see the little pink dots. These are all the amino acids that we're highlighting in our protein. So just drag the scroll bar over to the right and then highlight more. So just make sure everything in the A subunit gets highlighted. Still have a little bit of ways to go. Highlight more of the A subunit. Keep going until we get to proline number 274. And then we're also going to include the two zinc ions because those are present in the A subunit as well of beta-lactamase. So when you finish selecting all these amino acids, you'll notice that a third row came up here that says SELE, which is the selected region. We're just going to rename that right now. We're going to go to the action button, so A, rename selection. We're going to change from SELE into, we're just going to call that A. You hit enter. And now that's your A subunit that you are selecting. Okay, the next thing we're going to do is check out how to change the color of the A subunit, because right now it is green and we want it to look a little bit different compared to the rest of the other subunits in the protein. So let's just change the color of that. Right now I have A selected, so you see this is deselecting it. This is selecting the A subunit. We're going to go to color, we're going to go to spectrum, and we're going to go to rainbow. And you can see this changes the color of the A subunit now to rainbow colors. Roy G. Biv. So now that we have the colors all selected as the rainbow, what we're going to do is we're going to start looking at a new selection. So first of all, we're going to just zoom in to the A subunit. So I just click on, so I selected my A subunit, go to action, go to zoom. We zoomed into the A subunit here. And now I'm kind of curious to look at just what atoms are involved in the active site only. So let's try zooming in on the zinc ions only. So what I'm going to do is I'm going to deselect the A subunits. I'm going to go look for my zinc ions, which are in the A subunit. So these two zinc ions over here. I'm going to select those. And as soon as you do that, you see that the new row gets made with the selected atoms, which are the zinc ones. And we're just going to do the renaming again. So I go to action, rename, selection. So change it from SELE to, let's just call this zinc, ZN. Hit enter. See these little two pink dots here represent the two zinc ions. I want to make those look a little bit more pleasing than just two dots. So what I'm going to do is I'm going to change that to spheres. So I'm going to go show, and then as, and then just scroll down here to spheres. And so those two red spheres are representative of my two zinc ions. I just want to change the color of them. So I'm going to go color, yellow, and now there are yellow spheres representing my zinc ions in the active site in the A subunit. And the next thing I'm interested in is the residues, the amino acid residues that are interacting with the zinc ions. So what I'm going to do is I'm going to go to action. I'm going to go to modify, around, and then residues within 4 angstroms, because those are the amino acid residues that are involved in binding those zinc ions. So we're going to do that. You can see that those are going to be the pink dots that represent the amino acid residue side chains that are interacting with the zinc ions. What I want to do is I want to zoom in to those. So I'm going to go action, zoom, and now we're zoomed into the active site region. The two spheres represented the zinc ions. These pink dots representative of the atoms involved in the side chains interacting with the zinc ions. So next I'm going to do to actually make those residues show up is I'm going to go show and then sticks. So I'm going to make them show up as sticks. And you can see now all of those residues that are interacting directly with the zinc ions in the active site. The next thing we're going to do is we're going to make those residues show up as different colors based on their atomic composition. So I'm going to go color and then by elements, and there's a second option here. So it shows you the different colors. So we have C, carbons are going to be green, hydrogens are going to be gray, nitrogens are going to be blue, oxygens as red, and sulfurs as orange. So I'm just going to click on that. And now you can kind of see exactly which residues are involved in binding those zinc ions. And you can tell which amino acids they are just by looking at that straight up, or you can also look in the amino acid sequence that's at the top of the screen here. So I'm just going to deselect that for now. So you can see that this would be, for example, this is an aspartic acid residue, that would be CH2. That would be the C double bond O to the other oxygen, the carboxylic acid group, so you can tell that way. The other way you can do it is just have your amino acid residue selected and just go along the bar here, and you can even tell exactly where in the peptide chain it's going to belong. So, for example, this histidine over here, right, the one I'm selecting and deselecting, this is histidine number 250, and as I go towards the left here, this would be, so this guy down here, this would be cysteine number 208, and then we have histidine 189, and just go down a little bit further, there's a histidine 120, histidine 122, and an aspartic acid 124, which is the one I was showing you before. So that's exactly how you can tell which residues are interacting with your zinc ions at the active site. And I pointed that out also in the PowerPoint file. All right. Next thing I want to do is just make this a little bit more cleaned up. We're just going to hide the main peptide backbone chain. So if I just want to look at the side chains, I don't want to care about the NC-alpha CO part of the peptide chain. I just go hide and then main chain, and that goes away. And now we have, if I just deselect this all, you can see that those are just my side chains interacting with the zinc ions. The last thing I want to do in this step here is just look at the fog in the background. So you can use your scroll wheel of your mouse. So if I move my scroll wheel forward away from me, what happens is you can see more of the protein background shows up, but that looks a little bit more messy. I can clean that up a little bit more by scrolling my scroll wheel towards me or going in the backwards direction. You can see that the proteins, the little background goes away, and I can just focus my attention on the actual amino acid residue side chains interacting with the zinc ions. Alright, so that's how I can focus in on looking at the active site, and that's how you can zoom in to look at some of the features of hemoglobin when you do your assignment. Okay, the next thing I'm going to do is I'm going to just look at the space filling model, or the surface of the A subunit. So I'm going to deselect everything that I have in the active site there. I'm going to go back to A, so that's my entire A subunit still selected there. I'm going to zoom, it's going to zoom out now towards just the A subunit. And the next thing that I want to do is I want to show what this whole A subunit is going to look like in a space filling model. So I'm going to go show and go to surface. Now make sure you don't make this mistake. Don't go show as surface, go show, and then don't go to as and just go to surface, because if you go to as, it overwrites everything that you just did in the internal part of the A subunit. You just want to look at the surface and not override whatever you just did with looking at the zinc ions and everything. So just go show surface, and there you go. You can see it's a space filling model now, and it kind of shows you the three-dimensional space that each of the atoms and all the different side chains occupy. And if you manipulate the figure a little bit and look around, you can kind of tell there's some holes here, and obviously that's not going to be happening in a real protein. The reason why they'll show up is because when they did the crystallography structure of, or the crystal structure of 3-SPU, what happened was that these little holes are the areas where the A subunit is attached to the B subunit, the C subunit, and for this project we're not really concerned about that. We just want to look at the A subunit. So to extract only the A subunit that I'm looking at, we're going to take the A here, I'm going to go to action, I'm going to go to extract object, and after you do that, there's a little hole there still, but for the most part my A subunit is now intact with no holes. And also you'll notice that a new row appeared in the control panel showing object 01, 1 of 1. Alright, since I want to focus all my attention on the A subunit, I'm just going to hide my 3-SPU, so whatever is not the A subunit. So I just go to my second row here, go to the H button, which is hide, and then hide everything. And now I just have my A subunit left over. It's pretty good at this point. So now we're just looking at the A subunit. When I want to make my final figures, I'm just going to make this look a little bit better, just in one color, and I'm going to look at the internal parts of the enzyme as well. So what I'm going to do right now is just save my session because I want to make sure that I don't make any more mistakes from here. So I'm just going to call this test 3, call whatever you want. And at this point now, I'm going to increase the beauty of my figure. Okay, so what I'm going to do is I'm just going to zoom into here one more time, and I'm going to change the background of my image. So I'm going to put that as white. And the rainbow color, as appealing as it is, I think it's usually in a publication or anything like that, you want it to be one color instead. So I'm just going to go, so I have my object selected right now, object 01, go to colors, go to blues, and then go to marine. So now I just have my A subunit present as one color, the marine color, and I'm just going to go and make the surface of this transparent so I can see the internal features, or if you want to call them the bones of the enzyme. So it's like taking a little x-ray of my A subunit. So I'm going to go to settings, transparency, surface, and then 80%. And you can see it gives a little shallow outline of your surface of your protein, and you can look at all the little internal features and look at the little active site there that you just included and did some work on. And now it looks, I think this looks pretty nice for a figure that you can put in a publication. Next thing we're going to do is we're going to identify some hydrogen bonds. So this particular protein doesn't have too many hydrogen bonds in the active site, but when you're looking at hemoglobin, you might find some more hydrogen bonds that you're interested in. So let's try identifying the hydrogen bonds in the active site. So what I'm going to do is, unfortunately, Pymol doesn't allow you just to select the active site anymore. So what I'm going to do is I'm just going to deselect. Oops, no, I don't want to do that. It's going to select the individual amino acids that are involved in the active site. So I'm going to go back to object 1. I'm going to recognize all the residues that are in my active site. So I'm going to go to His 120, His 122, aspartic acid 124, and scroll down, go all the way to histidine 189. I need cysteine 208 and histidine 250. And I'm also going to include the 2 zinc ions. Those are everything that I want in the active site. So you notice everything. So what Pymol did is made a new row. All my selected parts are now part of the active sites. And I'm just going to rename that. So change SELE to, I'm just going to call this active for active sites. So this is my active site region that's going to be selected over here. The next thing I wanted to do is find my hydrogen bonds that are present in my active site. So I'm just going to zoom in here. Going to go to action. Going to go to find and then polar contacts. And with selection, so you can see now Pymol has drawn in my hydrogen bonds. Those are my dashed yellow lines there. You can see that this particular structure doesn't have that many hydrogen bonds, but when you do hemoglobin, you're going to probably find quite a few more. So don't worry too much about this one, just know how to figure out where the hydrogen bonds are, and you'll notice that in the figure it created a new row so you can select or deselect those hydrogen bonds in your structure. Alright, next thing we're going to do is we're going to try overlaying some structures here. So what I'm going to do is I'm going to zoom out. So I'm going to go to my object 01, zoom out again. And we're going to do an overlay. So we're going to compare my 3-SPU structure to another beta-lactamase isolator from Pseudomonas and see if it matches up. See what I found with 3-SPU. So I'm going to go back to File. I'm going to go Open. I'm going to go to my downloads, and I'm going to open up 1KO3, which is the other beta-lactamase protein that's isolated from a different bacteria. So we're going to overlay the structure. So to do that, what you have to do is you need to go back to your control panel and make sure that you only have those two proteins selected. So I only want my object 01 selected. I don't want the hydrogen bonds. I don't care about the entire structure. Don't care about all. I just want my object 01 and my 1K03 selected. And once they are both selected, I'm going to put them together. So I'm going to take my 1K03 one line, go to action. I'm going to go to align, and then to molecule, object 01. And there you go, everything is now aligned and together, so they're overlaying on top of each other. But that's not too useful because right now the 1K03 structure is still present as lines. I want them present as cartoons. So I'm going to go to my 1K03 selection over here, and I'm just going to make that show up as a cartoon instead. And now you'll notice that all the different secondary structure elements are overlaying on top of each other in my figure. And now it looks kind of nice that I can just zoom in there one more time. And now I can use this as my final figure for my assignment because I'm showing all the differences between the two different proteins. Like, see how this alpha helix just lies a little bit further to the right compared to this one. So the one that's in the marine is my 3SPU. The one that's in the teal is my 1K03. So you can see, you can do some comparisons that way. The last thing is I'm just going to retexture my drawing just to make the drawing just look a little bit nicer. So I'm just going to go to setting, and I'm going to go to rendering, setting, rendering, texture, and go to Mate 2. And once I do that, it just looks a little bit shinier. But there you go. So that's what you can do with Pymol, at least for the beta-lactamase. You can do the same kind of things for hemoglobin after you've done your research and figured out exactly which residues are involved in the active sites, what the mutants are doing, etc. And hopefully this tutorial helps you out.